Good morning, everyone. Welcome to this Workshop 30. This is the Epidemiology and Management of CF Airways Infections. On behalf of myself, Mike Parkins, and Dr. Katie Heisert from the National Jewish in Denver, I'd like to welcome you to this session. Neither Dr. Heisert nor myself have anything to disclose pertaining to today's session. We're going to be talking about two themes in this session, epidemiology and management. From an epidemiology perspective, we have identified several interesting talks. What's paramount to today's session is to understand the changing nature of the airways pathogens in CF. And to my knowledge, this is the first session in a long time where we've had more non-Pseudomonas aeruginosa presentations and Pseudomonas aeruginosa presentations, which reflects the significant changes we've seen in Pseudomonas aeruginosa prevalence over time. And of course, the older our patients get, and the more treatments they receive, the more likely we're to be managing complex infections and treatment-resistant infections. Infection control, of course, is paramount in CF. And we're going to be presenting studies examining new tools in order to understand past transmission events to try and prevent future ones. From a management perspective, it's critical to remember that patient outcomes very poorly correlate with traditional in vitro susceptibility testing in CF, and that we're always looking for ways and means to identify new models and new biomarkers to predict success. And unfortunately, increasing resistance is a fact of life in CF as a result of significant treatment experience of our patient population. And we always are looking for new approaches to infection management and to better understand treatment response. And it's in this light, we're happy to welcome today's five speakers. We have Dr. Anna Clara Galdino from Cedar Mount Sinai, presenting on the molecular mechanisms of cephederocol resistance in Pseudomonas aeruginosa, cephederocol being a first-in-class siderophore-linked cephalosporin looking to co-opt bacterial iron scavenging systems to improve antibacterial activity. Juliana Svischuk from the University of Calgary looking at the prevalence of Staph aureus with inoculum-related resistance to beta-lactam antibiotics. Dr. Lauren Gonzalez looking at adaptive responses of Staph aureus to trimethoprim sulfamethoxazole. And then two talks from the National Jewish in Denver, Dr. Jerry Nick presenting a case study on a young man with advanced CF lung disease who was treated with combination antimicrobials and a phage cocktail targeting his Mycobacterium abscesses, looking at how that population changed over time. And Dr. Jane Gross, also from the National Jewish, presenting the HALT Ntm data, looking at healthcare-associated links related to non-tuberculous mycobacteria transmission among CF patients, combining both genomics and traditional epidemiologic investigations. Dr. Heisert's asked me to remind the audience that the Best Abstract Junior Investigator competition is on Wednesday, November 17th at 11 o'clock. Please pay attention to that. And with that, I'd like to transfer over to today's speakers.
Good morning. I'm Anna, and I'm a postdoc at George Lab at Cedar Sinai Medical Center. And I'm here today to tell you about the mechanism of cephederocol resistance in Pseudomonas aeruginosa. But before I begin my presentation, I want to thank the organizers for setting up this conference and for inviting me to speak. Also, there are no relationship to disclosure related to this presentation. Well, when Pseudomonas colonize the CF airways, the bacteria faces a unique combination of selective pressures, which include the pressures from the host immune system, a gradient of oxygen and nutrients, and the presence of high concentrations of different antibiotics. And as the time goes by, the bacteria evolve to become more adapt to this new environment. And Pseudomonas tend to become less virulent, to grow as a biofilm, and to become multi-drug resistant. And because of that, the management of Pseudomonas infection is a big challenge. So the development of new drugs to treat CF chronic infections is always a great demand. And in this scenario, cefiderocol comes as a great option. Cefiderocol is a new drug that was developed by Shionogi Company, and it was recently approved for treating urinary and lung infection. And unfortunately, it was not approved for treating CF-related infections yet. However, it's being used as a compassionate treatment for controlling CF infections without any other treatment option. And overall, cefiderocol was able to control these exacerbations, and it was improved the patient's clinical settings. And we can correlate the success of cefiderocol to its unique mechanism of action. Cefiderocol is a cephalosporin, which has a catechol group that binds iron that chelates iron from the environment. And once it's bind to iron, cefiderocol takes advantage from bacterial transporters to get it inside cell. And in Pseudomonas pyoe is the protein responsible for actively import cefiderocol from the extracellular environment. Then, inside cell, cefiderocol inhibits the cell wall synthesis by targeting penicillin binding protein like any other cephalosporin. And when it comes to the potential mechanism of cefiderocol resistance, it was previously reported that the disruption of pyoe leads to a hefty increase of cefiderocol resistance level. However, mutations on pyoe does not entirely explain the cefiderocol resistance. So the aim of my project is to identify and characterize new molecular mechanisms of cefiderocol resistance in Pseudomonas aeruginosa. Then, intending to identify mutations that arises upon cefiderocol treatment, we perform an evolution experiment, where we initially pre-adapt the lab strain PA01 for 10 days in a CFM2. And this media mimics the chemical composition of CF1s. Then, after that, we propagate 8 different lineages of those pre-adapt population with increasing concentrations of cefiderocol. And we did this experiment under 2 different lifestyles. The first one is the planktonic, well-mixed population, and the aggregate biofilm models, where the bacteria grow in CF media with a little bit of agar that allows the bacteria to grow as a small aggregate. And that model reproduced the structure and the drug gradient and the nutritional limiting conditions that bacteria encounter when it's growing in CF1s. And when the population finally achieved growth at very high concentrations of cefiderocol, we harvest the cells and we sequence the whole population. Initially, we observed that cefiderocol resistance evolved as a stepwise pattern, which is an evidence that the resistance, the cefiderocol resistance phenotype does not rely on only one mutation. Instead, many mutations act together to lead Pseudomonas to resist even at very high concentrations of cefiderocol. Then we also observed that it takes around 20 days for aggregates population to grow under the highest concentration of cefiderocol, while it takes only 12 days for planktonic populations to grow at the same concentration. And this difference might reflect the drug gradient that aggregate cells faces, and also it's the slower metabolism that aggregate cells can have. And then we next look directly to the mutations we detect in the end of our evolution experiment. And surprisingly, we observed that all evolved populations were genetically diverse, exhibiting several mutations at intermediate frequencies. And here on this table, we summarize the mutations we detect in more than 2 populations, both for planktonic and aggregate evolutions. And each column represents one population, and the rows represent the mutations we detect. And the color gradient is how frequently these mutations were observed within a population. And among the populations we identify, we observe that mutations in several genes related to iron acquisition system, including mutations in PVDT, which is responsible for pyoverdin recycling, and PCHEF, PCHE and PCHF, which are 2 enzymes from pyochylin biosynthesis pathway. Also, we found different point mutations in CPXS, both in planktonic and aggregate populations. And CPXS is a sensor for envelope stress, and its correlate molecule, the CPXR, was reported as a MAX-AB regulator, which can tell us that cefiderocol can be a substrate for MAX-AB. And finally, we observed across 6 different aggregate populations a large deletion of DNA that eliminate 23 genes, which includes genes related to antimicrobial resistance, such as MAX-T, MAX-EF, and OPRN. Then, to test whether these mutations can lead to cefiderocol resistance, we perform some susceptibility tests on transposon mutant. And comparing with the wild type and the negative control for transposon insertions, we observed that the disruption of iron acquisition systems, here highlighted in yellow, leads to an increase of cefiderocol resistance levels, while it does not affect the cefepime and ceftazidime susceptibility. Then we next want to investigate how does cefiderocol resistance affect the bacteria's fitness. For that, we perform an in vitro competition assay in the absence of cefiderocol. In this experiment, we tagged the ancestor with MAPL, fluorescence protein, and with and the evolved population with YFP. And then we let them grow together at the same ratio. Then we follow the fluorescence signal, and we calculate the competition index. If the competition index were more than 1, it means that evolved population is winning. However, if the index were less than 1, it means that the ancestor is winning. And here we can observe that in the absence of the drug, the ancestor population outcompetes against the majority of evolved population. In addition, we use confocal microscopy to observe this competition in aggregate. And here, in this top bottom and this side view, we can clearly see that the ancestor population in red could form bigger aggregates when we compare to the evolved population. And from this data, we can infer that cefiderocol resistance comes with a fitness cost. And then we asked ourselves what happened with the bacteria fitness under cefiderocol pressure. So we did the same experiment, but now in the presence of high concentrations of cefiderocol. And here we can see that almost all most of the evolved population outcompete over the ancestor, which is kind of expected since those populations were under cefiderocol pressures for many, many days. However, we were very curious because we saw that two evolved populations promote some protection for ancestor population, allowing them to grow even in high concentrations of Cephiderocol at similar growth of the evolved population. And more interest, we observed the same protective actions on aggregate populations, where the sensitive cells in red were shielded by the evolved population, the evolved and resistant population in green. And so far, we do not know what drives the protective competition between Cephiderocol sensitive and resistant population. But we are very excited to address these questions. And we're planning to use single cell RNA techniques to identify how those resistance populations protect the sensitive ones. And just to quickly summarize my presentation today, we observed that Pseudomonas become Cephiderocol resistance in vitro, and the resistance phenotype is multifactorial, and the disruption of iron existence systems leads to an increase of Cephiderocol resistance. And these mutations that leads to Cephiderocol resistance pay a fitness cost. And interestingly, Cephiderocol, some Cephiderocol resistant population can protect the susceptible ones. But the main takeaway from my presentation is that the utility of Cephiderocol in chronic CF infection is still under investigation. However, this is to provide a great information that can help the CF community to improve the usage of Cephiderocol, aiming to delay the spread of Cephiderocol resistance. With that, I wanted to acknowledge our financial resources, and I would say a big thank you for all my lab mates from George Lab. It's been great work with them, and without them, this project would not be possible. And finally, I would like to thank you so much for your attention, and I'll be more than happy to take any question you might have during the Q&A section. Thanks.
Excellent. Thanks for that presentation, Dr. Galdino. We have a few questions for you. Question one, was resistance of aggregates tested with bacteria still in aggregates, or were aggregates dispersed before testing resistance? The rationale being exopolysaccharide production by Pseudomonas has been shown to contribute to antibiotic resistance in grown aggregates.
Thanks for the question. This is a really good question. Actually, we test the Cephiderocol resistance with the aggregates not structure. So because we collect the cells, and after that we check the MICs using the standard media, which is not the media we use for evolving the populations, because Cephiderocol is highly dependent on iron concentration. So we use the iron depleted millihinton for checking the MICs. But yeah, it makes sense. Like, of course, we expect that under aggregates, the MIC would be even higher than what we detect.
Can I ask as well, you presented data on ceftazidime and cefepime resistance amongst your evolved population. Did you look at any non-beta-lactam related drugs? The rationale being many of your mechanisms related to outer membrane permeability and efflux.
Oh, that's a really great question. I also check the MICs for other, in my evolved population, I check the MICs for other drugs, not beta-lactam drugs. And based on what I remember from the top of my head, I remember that the evolved population exhibit high MICs for all the other beta-lactams, but it also have a slightly increase on colistin and polymyxin B.
Thank you. Is there a role for testing development of resistance in an in vivo model, and might evolution be different in host tissue?
That's a really great question. Thank you, though. I suppose that the evolution in host is different. It could be different. So for confirm that all the mutations we detect in our in vitro experiment, we expect to acquire some clinical isolate that confirm that from patients that receive cefiderocol and the treatment was not efficient, to compare like if the mutations detected on that strains is also, it matches with what we detect. And regarding to the evolution in vivo model, there's a paper that was developed by the Shionogi company that shows that the in vitro evolution does not correlate with in vivo evolution performed with Stenotrophomonas. But yeah, it's possible. It's just a matter of planning.
The next question, is it known if there's synergy between cefiderocol and other antibiotic classes?
There's a few papers that show some synergistic effect with cefiderocol and some bacteriophage therapy. But other than that, I'm not completely aware about those combinations. And also we did a very primarily data that we combine pyoverden and pyocolin and cefiderocol, and we did a checkerboard assay, and interestingly, pyocolin, the combination of pyocolin and cefiderocol was highly synergistic. So we believe that the deletion that we observed on the pyocolin biosynth pathway is really critical for cefiderocol resistance.
Excellent. Well, thanks for a great presentation today, Dr. Ogaldino.
Thank you.
Hello, my name is Juliana Swischuk. I'm currently a master's student at the University of Calgary in Calgary, Alberta in Canada. Thank you so much to the moderators for having me here today. It's an absolute privilege to be able to present to you the work of my undergraduate research thesis. Now, the title of my talk today is The Prevalence of Staphylococcus aureus in Cystic Fibrosis with Reduced Susceptibility to Beta-lactam Antibiotics at High Inoculum. And please note, I have no relationships to disclose related to this presentation. So as for a little bit of background, the Canadian Cystic Fibrosis Registry recently reported Staphylococcus aureus as currently the most prevalent pathogen isolated from the airways of individuals with cystic fibrosis at about a prevalence of 53%. And this data is fairly consistent in countries beyond just Canada. So what's very interesting about Staph aureus is that its phenotype can actually influence its pathobiology. And we see this in cases such as methicillin-resistant Staph aureus or MRSA, small colony variants, and even Panton-Valentine leukocidin. Now, although many studies have emerged on the topic of MRSA, especially in cystic fibrosis, MSSA or methicillin-sensitive Staph aureus, albeit it being the more prevalent form of Staph aureus, is comparatively understudied relative to MRSA, even in the context of cystic fibrosis. Now, when we're talking about Staph aureus, something really important to note is the BLAZ gene, and its regulon is shown in the upper right-hand corner here. Now, BLAZ is essentially the causative agent of Staph aureus penicillin resistance, and it's identified in around 90% of all Staph aureus isolates, so it's fairly common. It encodes the BLAZ protein, which is a beta-lactamase that acts to hydrolyze the very reactive beta-lactam ring that we see in all beta-lactam antibiotics, effectively deactivating the antibiotic as a whole. Now, it can be subdivided into 4 different subtypes, A, B, C, and D, each characterized by a different kinetic or substrate profile in its protein form, though genotypically all subtypes are very similar. Now, the critical thing to note here is that aside from simple penicillins such as aminopenicillins and ureadopenicillins, Beta-lactams were previously thought to be entirely resistant to the effects of Blazed. However, as we'll come to find through the progress of my talk, this might not necessarily be the case. And so the premise of my work lies in something called the MSSA inoculum effect. Now, over the past many, many years, it's been noted that some MSSA isolates can actually produce sufficient amounts of that Blazed beta-lactamase to hydrolyze otherwise resilient antibiotics. But this can only occur when the pathogen is present at high concentration, or above 10 to the 5 colony-forming units per milliliter. And this is called the inoculum effect. Now, 10 to the 5 is what we typically see in the clinical laboratory. So when we receive results from the clinical lab, these are how the bacteria is acting at a standard inoculum. We're not sure how it's acting at high inoculum, and this can be very different. Now, this inoculum effect is applicable to high inoculum infections, of course, such as in the case of osteomyelitis, endocarditis, and bacteremia. And of course, cystic fibrosis has a very high inoculum nature in terms of lung infections associated with CF. However, the inoculum effect has never been studied in the context of cystic fibrosis. If I draw your attention to this table here from an article published by Fisher earlier this year, we can see that amongst pathogens isolated from cystic fibrosis sputum, both MRSA and MSSA exist at inocula above 10 to the 5, reaching 10 to the 6, and even sometimes 10 to the 7. Now, furthermore, the inoculum effect has mostly been studied in the context of the first-generation cephalosporin, cefazolin, as seen here, and very little has been done in the area of other antibiotics. And of course, as we know, cystic fibrosis, in terms of lung infections, is very commonly a polymicrobial disease. And so we're oftentimes looking at drugs beyond just cefazolin, broader spectrum antibiotics, when thinking of treatment for cystic fibrosis. And so we have very little to go off of when looking at inoculum effects in CF. So like I mentioned, many studies have focused on the cefazolin inoculum effect, and this has particularly been the case in bloodstream infections. Now, these studies have reported prevalence rates from about 16.4 to 4.4% and 4% to 54.5%, and these two are indicative of two separate definitions that are very commonly used when looking at inoculum effects, and I'll get into this as we progress in the talk. So what's most important for me to pinpoint here is that the cefazolin inoculum effect has been found to correlate with reduced bacterial killing, decreased treatment efficacy, and increased patient mortality in those with bloodstream infections. Now, once again, looking at this table here from a paper published by Nannini and colleagues in 2009, we can see that when MSSA was tested with cefazolin at high inoculum, we see this trailing edge of isolates that are resilient to cefazolin at concentrations well beyond the CLSI breakpoint for non-susceptibility. And so this is exactly what we're interested in. How is this acting in cystic fibrosis, and what are the implications of the inoculum effect? And so for our objectives, we set out to determine the prevalence of inoculum effects in methicillin-sensitive Staph aureus isolated from CF sputum with cefazolin and the clinical gold standard isoxazolyl penicillin, cloxacillin. Furthermore, we wanted to explore inoculum effects in other CF-relevant antibiotics beyond just cefazolin, including cefepime, meropenem, ceftazidime, and piperacillin-tazobactam. And of course, we wanted to examine the molecular basis of inoculum effects through Blazed typing into those subtypes of A, B, C, and D to see whether we can find any correlations or associations between inoculum effects and Blazed type. Now, we hypothesized that the inoculum effect exists in CF sputum-derived MSSA and is in fact correlated to the presence of specific alleles of the Blazed gene. Now, for our methods, we started out by screening all 220 adults attending the Calgary Adult Cystic Fibrosis Clinic. Our only inclusion criteria was an underlying diagnosis of cystic fibrosis, and we essentially screened all these 220 patients to see which of them grew Staph aureus over the course of 2013 to 2016. And we found 98 patients who grew Staph aureus at least on one occasion. Some patients grew it more than once, and some grew it only once. For those who grew it more than once within 1 year, we only selected one sputum sample per year for a total of 238 MSSA isolates. Now, we didn't exclude any isolates that we got, nor did we exclude any patients. So we then went over to streak out the MSSA that we got onto tryptic soy agar and create standard and high inoculum bacterial suspensions in diluted saline. Now, we defined standard inoculum as the typical 10 to the 5 CFU per milliliter and high inoculum as 10 to the 7 CFU per milliliter. The first isolate from each patient, so 98, was tested with our 6 antibiotics of interest, whereas the remaining isolates, the remaining 140, were tested only with cefazolin, meropenem, and piperacillin-tazobactam, and I'll get into why we chose these in particular as I go on. Lastly, BLAZ typing was performed using standard PCR for the BLAZ gene on the first isolate of each patient, so once again, that 98. And gene typing was performed using the Comprehensive Antibiotic Resistance Database, or CARD, as well as BLAST alignments. So for our first bit of results here, I'd like to draw your attention to the two standard definitions that are used when observing inoculum effect studies. So definition 1, or what's called simply the inoculum effect, is defined as an isolate being positive for it if there exists at least a fourfold difference between the minimum inhibitory concentration, or MIC, between standard and high inoculum. So minimum inhibitory concentration is essentially the lowest concentration of antibiotic required to inhibit visible bacterial growth. Contrarily, definition 2, or the pronounced inoculum effect, is defined as an isolate being positive for it if an isolate is susceptible to a drug at standard inoculum, but not susceptible at high inoculum. And so looking at this table here, we have our antibiotics of interest on the left-hand side and minimum inhibitory concentration breakdowns here horizontally. Now I've compared the MIC50 and MIC90, or the 50th percentile of MICs and 90th percentile of MICs for all 238 isolates tested against these antibiotics. I've compared them in terms of standard and high inoculum. And what's important to note right off the bat is that in some of these drugs, we can see an increase in both MIC50 and in MIC90. And I'll go into this more on the next slide. This is particularly relevant for drugs such as cefazolin and piperacillin-tazobactam, but we'll come back to this. Furthermore, when we're looking at inoculum effect prevalence, we see a very diverse range of prevalences across the drugs, and more so, we see a far greater amount of inoculum effects when compared to pronounced inoculum effects, again, most common in cefazolin and piperacillin-tazobactam. However, given that we do see more prevalences in our inoculum effect as opposed to pronounced inoculum effect, we decided to move forward with only focusing on inoculum effects by definition 1. And so, as I mentioned, when approaching this MIC distribution when looking at cefazolin and piperacillin-tazobactam, at standard inoculum, we note this very typical distribution here with our breakpoint for cefazolin resistance being at 16 micrograms per milliliter. We have our minimum inhibitory concentrations here on the x-axis and the percent of MSSA isolates from our cohort who exhibited that MIC. So when we look at standard inoculum, we see an MIC50 of 0.5 micrograms per milliliter, and MIC90 at 1 microgram per milliliter. However, when looking at high inoculum, we see the significant rightwards shift of MIC distribution, with MIC50 at 1 microgram per milliliter and MIC90 at 4 micrograms per milliliter for high inoculum, a fairly significant rightwards shift. Now, when looking at piperacillin-tazobactam, it's important to note that we kept tazobactam concentration stable as we looked at these different MICs, but we varied piperacillin concentration. And so at standard inoculum, once again, we see a fairly standard MIC distribution here, with MIC50 being at 4 micrograms per milliliter and MIC90 being at 8 micrograms per milliliter. But we see this very drastic shift to the right when looking at high inoculum MIC distribution, with this trailing end of isolates that are completely resilient to the drug well beyond its breakpoint for resistance at 16 micrograms per milliliter. And this is in fact indicative of that pronounced inoculum effect that we saw in the piperacillin-tazobactam. Also, as we can see, MIC50 shifts from 4 to 8 micrograms per milliliter between inocula, and MIC90 shifts from 8 to 128, once again showing a drastic rightwards shift and a drastic difference. And this is particularly concerning because piperacillin-tazobactam is very commonly used when looking at cystic fibrosis, where MSSA infections tend to very commonly exist at high inoculum. And so to summarize this little bit of data here, we found out that cefazolin, of course, and piperacillin-tazobactam show prominent inoculum effects. And so moving forward, we paid a little bit closer attention to those drugs in particular. So as for our molecular analysis of BLAZ types, when looking at our 98 MSSA isolates, so the first from each patient, we found that 24 were negative for the BLAZ gene, and as expected, none of them showed the inoculum effect, whereas 74 were positive for the BLAZ gene. And we see this fairly diverse distribution of BLAZ types, independent of whether they were inoculum effect negative or inoculum effect positive. And so looking at this a little bit closer, we noted that isolates exhibiting the piperacillin-tazobactam inoculum effect very commonly had type C BLAZ, whereas isolates showing the cefazolin inoculum effect very commonly had type A BLAZ. And so looking at this a little bit more closely, we were actually able to find that these results are statistically significant. So using a Fisher's exact test, we were able to compare isolates that showed the cefazolin inoculum effect and were positive for BLAZ type A, whereas those that were not type A, they showed a statistically significant difference between the two cohorts. And the same thing can be said for Piperacillin tazobactam, but in the context of BLAZ type C versus non-type C, we saw a statistically significant difference. And so we were able to conclude that type A BLAZ is statistically associated with the cefazolin inoculum effect, while type C was associated with the Piperacillin tazobactam inoculum effect. And so in conclusion, we were able to find that the inoculum and pronounced inoculum effect exist only in BLAZ positive isolates from individuals with cystic fibrosis. Furthermore, the inoculum effect was commonly found with cefazolin at about 19% and Piperacillin tazobactam at about 42%. And this could possibly impact patients' potential for improvement with treatment. If we're treating patients who have inoculum effect positive MSSA infections and we're treating them with inoculum effect relevant antibiotics, how effective is that treatment really going to be? Lastly, we found that type A BLAZ did associate with the cefazolin inoculum effect, and type C BLAZ associated with the Piperacillin tazobactam inoculum effect. As far as applications and future directions go, we're currently looking into how does the inoculum and pronounced inoculum effect relate to short and long-term patient outcomes. Now, more specifically, we're looking at parameters such as pulmonary exacerbation treatment recovery, lung function decline, pulmonary exacerbation frequency, and so on. Now, we're looking at whether these parameters tend to worsen when patients are infected with inoculum effect positive MSSA strains, and how do these parameters vary when we're treating patients with inoculum effect relevant antibiotics. Furthermore, we want to know how should the inoculum effect and pronounced inoculum effect be considered when establishing treatment plans and assessing for response? Is there a way for us to incorporate inoculum effect testing into standard of care practices where we're able to use inoculum effect phenotypes as a means of guiding how we choose to treat patients who might have this phenotype as an infection? So thank you so much once again for taking the time to listen to my talk. Thank you also to my supervisor, Dr. Michael Parkins, and my undergraduate co-supervisor, Dr. Douglas Story, for their endless support. Thank you so much also to the Parkins Lab, particularly Barbara Waddell, for their support and guidance. Thank you, a special thank you to many of the faculty members of the University of Calgary at the Cummings School of Medicine, and of course, Dr. Sheila Wang for providing some of the isolates that we used as controls in our study. Thank you so much. I hope you enjoyed my talk, and I wish you a wonderful rest of your day. Thank you.
Thanks for your presentation, Juliana. We have several questions from the audience. Question number one, it looked like there was no inoculum effect with meropenem. As a clinician, if I need empiric antibiotics for a patient growing both Pseudomonas and Staph, should I pick meropenem?
Thank you for your question. Yes, so meropenem has been not only tested by us in terms of inoculum effects, but by other studies with fairly low, if not complete absence of inoculum effect relevant presence. So I think meropenem would be a strong antibiotic to use for sure. Whether it applies to a vast variety of applicable infection types, that's something that, given that I'm not a clinician, I wouldn't be able to say. But based on data present to this day, meropenem seems to show the least amount of inoculum effects that we're able to see. So it's a pretty strong contender in that sense.
Next question. Do you know if inoculum effects changed in polymicrobial airways infections?
Yeah, that's a wonderful question. We're in the process of looking at that, actually. We have data available from this cohort here where we are able to see what kind of infections patients were infected with at the time of isolating these inoculum effect relevant strains, as well as beforehand. And so we're wanting to see whether patient outcomes change in relation to whether they are co-infected with certain pathogens like Pseudomonas or not. So that's what we're currently trying to establish, but it's a wonderful question and it's definitely where we're going.
Excellent. Next question, Juliana. You've shown that the inoculum effect for cefazolin associates with the BLAZ A and tasis in the BLAZ C. Are there other Staph aureus factors that predispose to the inoculum effect?
Yes, so inoculum effects there's a broad category of inoculum effects. BLAZ is not the only one. Furthermore, inoculum effects have been tested beyond just staph as well, but there are classical signs of inoculum effects, excluding just enzymatic degradation. Just the bacteria existing at a higher inoculum has a lot of elements to it that give it this quality of having the potential for decreased susceptibility. Less antibiotic interacts with the bacteria in the first place per unit cell. Pathogens are more likely to reach stationary growth phase faster. And so you have a decreased rate of expression of different antibiotic targets at that point as well. So there are factors definitely beyond just BLAZ that have been noted in inoculum effects for sure, yes.
Excellent. We have a couple of questions from Dr. Luke Hoffman. Fascinating results and great presentation. Do you know whether the inoculum effect phenotypes or BLAZ genotypes vary among isolates within an individual patient's samples?
Yeah, it's a wonderful question. Another one that we're literally, as we speak, looking through. So if I understand your question correctly, we have from one patient, we're able to have access to data from a number of years. So if a patient, say at one point in time, had a MSSA inoculum effect relevant isolate, if they had another MSSA inoculum effect positive isolate, are they the same strain? We have pulsotype data available on that, so we're able to see whether this effect is maintained over time. If it's not the same strain, is there something in the patient's demographic that's allowing this acquisition of this strain more so? So we're currently in the process of looking into that as well. It's a fantastic point to consider.
Another question from Dr. Hoffman. Do you know whether commonly seen susceptibility phenotypes like borderline MICs or heteroresistance have any relationship with the inoculum effect presence?
Yeah, so as far as I understand, borderline resistance is a slightly different thing than inoculum effect resistance. Borderline oxacillin resistance has actually been considered in the context of cystic fibrosis with some very, very interesting results, actually. But from what I can tell, borderline is more so associated with the intrinsic nature of the pathogen itself, whereas inoculum resistance specifically bases on the bacterial density that's present at that time. So at a standard inoculum, we don't see high inoculum resistance, but whereas borderline resistance, I think, can still be observed. So they're slightly different in that sense. I wouldn't know of any relationships between them, no.
Excellent. Thanks for answering such great questions from the crowd, as well as a great presentation. Thanks, Juliana.
Thank you.
Hello, everyone. I am Lauren, a PhD student at the University of Washington here in Seattle. And today I'm going to be talking about our work with characterizing the adaptive responses of Staph aureus to trimethoprim sulfamethoxazole. Before I go on, I want to thank the Cystic Fibrosis Foundation, as well as the organizers, for setting up another wonderful virtual event. Regarding this presentation, I have no relationships to disclose. So as many of us know, staph is a common commensal organism in our everyday lives. In the anterior nares, up to 60% of us can be intermittently carrying staph. And while it acts as a commensal and is often carried asymptomatically, it's also a major opportunistic pathogen and the leading cause of several infections, such as bacteremia, skin and soft tissue infections, and respiratory infections. One particularly concerning facet of staph's capabilities as a pathogen is its ability to establish chronic infections that can often be difficult to eradicate. People with the genetic disease cystic fibrosis, or CF, are particularly at risk for staph infections. One of the characteristics of CF is the buildup of a thick mucus that's hard to clear, which results in chronic infection. When you look at the prevalence of respiratory microorganisms, as reported by percentage of individuals over the past 2 decades, you see that staph and its subtype methicillin-resistant Staph aureus have increased in prevalence, making staph a relevant pathogen to study. While the role of staph in CF lung infections is not entirely clear, studies show that in people with CF, persistent Staph aureus infection is associated with increased lung inflammation as well as decreased lung function. When it comes to treating these infections, antibiotics really do serve as the cornerstone of care. So while there are many antibiotics used in treatment, the one I'm going to be talking to you about today is the combination antibiotic trimethoprim sulfamethoxazole. You'll see this abbreviated as SXT, but you'll hear Trimsulfa. Now, this is an effective anti-staphylococcal and especially when other treatment options are limited, and its efficacy hinges upon its ability to inhibit folate metabolism in bacteria. So what exactly does trimsulfa do? Separately, these two drugs are bacteriostatic, meaning they only pause the growth of Staph aureus, but together they work synergistically. For the sake of time, I'm not going to go over the entire folate pathway, but what's important to take in is that it inhibits two steps of the folate pathway, which ultimately shuts off the production of tetrahydrofolate. Now, tetrahydrofolate is an important cofactor in the synthesis of amino acids, carbohydrate metabolism, as well as nucleotides, particularly particularly thymidine. As you can see here, it's responsible for the conversion of deoxyuridine monophosphate to deoxythymidine monophosphate, which is then modified and incorporated into DNA. So, without the production of tetrahydrofolate, thymidine is not produced. And at this point, trimsulfa effectively starves the cell of thymidine. And this particular starved state is known as thymineless death. It results in the death of the cell. While there's a lot we know about thymineless death, there's also a lot we don't know. And current literature points to the accumulation of reactive oxygen species, as well as DNA lesions and misrepair and DNA breaks that ultimately result in death. So trimsulfa is an effective way to kill a pathogen. However, the efficacy of trimsulfa, like many antibiotics, is limited by mutational resistance. And in staph, this often occurs through a loss of function mutation in thymidylate synthase. This enzyme is responsible for the conversion of DUMP to DTMP along with tetrahydrofolate. And this particular mutation has been well documented in both the laboratory and clinically. So without this enzyme, thymidine is not produced and not incorporated into DNA. And that's something that was going to happen with Bactrim eventually. However, what's unique about this situation is that with the uptake of exogenous thymidine, only with the uptake of exogenous thymidine, Staph aureus is actually able to avoid thymineless death. So essentially, thymidine serves as an antagonist to trimsulfa action. So to summarize what we've gone over so far, without exogenous thymidine, trimsulfa, which inhibits the folate pathway, induces thymineless death. However, we know that treatment doesn't always lead to eradication. And we know that thymidine can antagonize trimsulfa action. And this last point really prompted a question for us. Is this happening in the lung? Is there sufficient exogenous thymidine in the airways to support Staph aureus survival? So we know that the bioavailability of certain compounds like thymidine in the environment can play a big role in survival. And up to this point, the levels of thymidine in sputum haven't been reported as far as we know. So after culturing for Staph aureus, we analyzed 53 samples via liquid chromatography mass spectrometry, where we used peak areas to determine concentrations of thymidine, as well as deoxythymidine monophosphate, diphosphate, and triphosphate. And what we hypothesized was that sputum would contain free thymidine levels and thymidine analogs at levels suitable enough for the growth of Staph aureus. It's not exactly what we found. What we saw was that levels of thymidine and monophosphate ranged from below the level, from the limit of detection to 13.5 micrograms per mil. When we looked at diphosphate and triphosphate, we saw that most of the samples were at or below the limit of detection. And so seeing low levels of thymidine in sputum prompted us to ask, well, is there a threshold that allows for survival? And is that what these low levels are showing us? And so what we did was we exposed Staph aureus Newman to trimsulfa over 12 hours. And we saw that at least 0.25 micrograms per mil was required for sustained survival in vitro. And so when we referred back to our samples, we saw that only 28% of our samples actually contained levels at or above this threshold. One unique thing that we saw, though, was that at the 24-hour mark, we actually saw an increase in culture density at some of these lower thymidine concentrations. Again, prompting another question, how is Staph aureus surviving at these low thymidine levels? We see low thymidine levels here in vitro. We see them at low levels in sputum. How ultimately is Staph surviving? We hypothesized that Staph survives through a pathway unrelated to biosynthesis, that there are unknown mechanisms that Staph is using. And so to further investigate this, we cultured out that 24-hour time point, and we extracted representative isolates for a mutational analysis. So what I'm showing you here is I'm reporting the gene that was mutated, the phenotype of the actual isolate, and then a predicted process that's impacted. And as you can see here in this chart, what we found was that most of the mutations were for genes that fed into the electron transport chain. Now, this leads us to question what the connection might be between folate pathway inhibition and the electron transport chain. We know that the electron transport chain is a source of reactive oxygen species in the cell, and that reactive oxygen species have been reported. Increased levels have been reported during thymineless death. So that led us to ask, well, are Staph aureus adaptive mutants surviving Bactrim pressure through during thymidine limitation through the reduction of ROS? So in order to investigate that, we used a fluorescent indicator that will fluoresce in the presence of reactive oxygen species. And we put this through a flow cytometer that read each event or cell and its fluorescence as reported like this. So what I'm showing you here are on the x-axis counts or cells, and on the y-axis fluorescence intensity. And we monitored this over 6 hours when active thymineless death is occurring. And as you can see at the 0-hour mark, we didn't really see a huge difference between treatment and no treatment in Staph aureus strain human. However, what we saw was at the 6-hour mark, we actually saw a shift in fluorescence peak indicative of an elevated level of intracellular ROS. When we looked at Thy A, essentially a phenocopy of Staph aureus treated with Bactrim, we also looked at over the same time period, and we saw a similar occurrence that at 6 hours, when thymineless death is actively occurring for Thy A in the absence of thymidine, we see an increase in reactive oxygen species. We next wanted to look at a few of our mutants. Now, our mutants that we used are in the JE2 background, and so we tested JE2 with Bactrim with trim sulfa exposure, and we saw a similar result: an increase in reactive oxygen species or a shift in fluorescence intensity. The mutant we decided to focus on was a Hep T mutant. Hep T is a hyperpigmented mutant, and this pigmentation is a result of staphyloxanthin overproduction. And literature has reported that staphyloxanthin actually aids in survival in staph when exposed to reactive oxygen species. So at 0 hours, we saw an overlap between the peaks, our baseline ROS levels. However, at 6 hours, what we saw was a minimal shift, a shift much less than what we saw with our parent strain exposed to trim sulfa. So just to summarize everything I've gone over today, we know that the mechanism of action of Bactrim is not well understood. Evidence suggests that trim sulfa induces thymineless death. However, it's unclear how Staph aureus survives. We reported that free thymidine levels in CF sputum are often too low to prevent thymineless death, and that adaptive variants that don't undergo thymineless death carry mutations in the electron transport chain, as well as some metabolic pathways. We also saw that Trim-sulfa can induce high levels of reactive oxygen species, similar to what was seen in the literature for E. coli. But this ultimately suggests a potential role for reactive oxygen species in trim-sulfa killing. However, whether reactive oxygen species contribute to cell death or reflect a general stress response is not clear, but it does suggest promising directions for improved anti-staphylococcal agents. Before I take any questions, I want to take a moment to acknowledge a few wonderful people. The Hoffman Lab has really played a big role in this project's progress, particularly Dan Walter. He's been instrumental. And then the Salaponte Lab has been an incredible source of expertise and knowledge throughout this project's progress. And last but certainly not least, I want to thank all of you for your time and your attention. And I am happy to take any questions at this point.
Thanks, Lauren, for a very interesting talk. We have a few questions from the audience. The first is, are there other therapies or antibiotics that could be combined with Septra to further enhance ROS?
Yeah, good question. We haven't explicitly tested this, but there is a lot of literature suggesting that ROS production is increased with the use of other antibiotics. So that's entirely possible that co-treatment could enhance ROS production.
It's really surprising to me that thymidine levels are as low as they are in CF airway secretions, given that dry weight of sputum is mostly attributable to neutrophil DNA. How do you explain that?
Well, one explanation we were thinking about is some of the samples that are Staph positive. We may be capturing it at a time point where thymidine has been consumed already. I mean, we only have limited samples that we've tested, so that's one explanation. But yeah, it's a very, very interesting dynamic that we weren't expecting.
Excellent. Next question is ROS in Staph solely within the bacterium, or does ROS made by Staph contribute to extracellular ROS?
Really good question. Based on what we've tested, we can really only speak to ROS produced within. It's entirely possible that there are extracellular, I mean, it is possible that there are extracellular sources of ROS, but we have not started to look at that just yet.
So question from Rafael Hernandez. Have you tested whether small molecule quenchers or antioxidants are able to promote survival of Staph when treated with TMP-SMX?
Yeah, awesome question. We're currently working on that. We have not tested that. Literature suggests that quenchers are gonna do help survival with trim-sulfa exposure. So that's definitely on the list.
Excellent. There's a question from Peter Jor. Did the ETC mutants have growth defects in the absence of Septra?
Yes. So metadione in human mutants had slight growth defects, but when treated with trim-sulfa, the difference between with and without treatment wasn't too different. So slightly compared to wild type.
Excellent. Thanks for a wonderful presentation and answering those questions for us, Lauren. Thank you.
Of course.
Hi, this is Jerry Nick, and I'd like to thank the organizers for inviting me to speak today. I do not have any relationships to disclose. This work was supported through the CF Foundation. So phage therapy is, of course, based on viruses that exclusively infect bacteria. And typically, these viruses enter a lysogenic cycle and do not damage the bacteria. But occasionally, viruses are found which enter a lytic cycle, and in that case, are capable of lysing the bacteria and then being released back into the milieu and infecting additional bacteria. So this technology is very appealing for the treatment of Mycobacterium abscessus, given the difficulty with antibiotic therapy. There's been a lot of interest in this, but actually there's only 2 reports available, and neither of them are in the CF airway. The first was a 15-year-old woman with CF post-transplant and disseminated Mycobacterium abscessus who did respond to therapy. The most recent one this summer was from an individual with non-CF bronchiectasis who failed therapy due to rapid development of antiphage neutralizing antibodies. Both of these were cases of M. abscessus subspecies Bacilliensis. So we do have some reports of phage for other CF pathogens, in particular Pseudomonas, and there has been a scattering of reports that indicate successful eradication. But also we've seen reports of the emergence of phage resistant by the Pseudomonas and other bacteria, as well as the emergence of new pathogens. So the objective of this study was to assess the effect of successful mycobacteriophage lysis on the genetic composition of a well-established Mycobacterium abscessus infection in the CF airway, with the hypothesis that successful phage treatment of abscessus will result in a decrease in genetic diversity within the persister isolates. So the case is a 26-year-old man with CF and treatment refractory abscessus. He has a rare mutation and was chronically infected with Pseudomonas and methicillin-resistant Staph. He has typical features of CF, including pancreatic insufficiency, diabetes, and malnutrition, and was infected with first positive cultures for abscessus in 2016. He was enrolled in the PREDICT trial and met diagnostic criteria in early 2017, and when therapy was started via the algorithm of the patient's trial. After a year of treatment, he was taken off protocol due to treatment failure and the need to expand the drugs being used. So isolates early on from his infection were sent to Graham Hatfield and Becca Diedrich in Pittsburgh, and they were able to identify 2 engineered phages that were lytic against his isolates, and that's shown by the assay here. The phage was administered through a compassionate use IND via the FDA following IRB approval and informed consent. We started treatment while he was in the hospital following the end of a typical exacerbation, and the 2-drug or the 2-phage cocktail was administered IV twice daily. He had been on intensive antibiotics and has continued on the antibiotics as well. And he has continued now for a year and administering the phage twice daily at home with weekly follow-up and labs, and monitoring his by cultures and radiographs per usual care. And this is a plot of his clinical course. Here the line is FEV1% predicted versus the date in days, with day 0 being the start of phage treatment. On the top, the hash marks indicate M. abscessus cultures, with the black hashes being negative cultures. The red hashes are positive cultures. And on the bottom bar, we see the start of antibiotics in black. And then he was approved about 90 days after start of phage therapy with highly effective modulator therapy. And I want to call your attention to the culture results after the start of phage. From day 0 to 96, 6 of his 7 cultures were positive. But from day 118 to 362, 1 out of 9 cultures were positive, with the only positive culture coming on day 245. So this is a a diagram of the genetic diversity of the isolates as analyzed by whole genome sequencing from 2016 to 2021 with SNP numbers and position used as a read mapping approach. This is work by Rebecca Davidson and Fanjai at National Jewish. And what we can see is here in orange was the first positive culture, but in gray, we see common recent ancestors. And we see the development of a number of lineages with a shift in towards three separate lineages, letter A, B, and C, after the initiation of antibiotic therapy. And then a further reduction in diversity shown here by the red line when the phage treatment was started. And we see here the persister isolate on day 245, which was quite separate from the earlier ones. But overall, the genetic diversity among this basically clonal infection was limited. There was only a total of 11 SNP difference across, even though there was several SNPs that were coding for key proteins. And this also allows us to estimate the mutation rate and the date of the initial common ancestor using a Bayesian evolutionary analysis. And from that, we can estimate that his initial infection was actually 30 months prior to his first positive culture, as shown on this plot, although there is a wide highest posterior density interval. We also were able to analyze the accessory genome, and that is demonstrated here for 50 isolates by hierarchical cluster analysis using Euclidean distant metrics, and each row represents a different gene. The presence of the gene is depicted in blue and the absence in light yellow. And we see the isolates ordered from earliest of the initial infection all the way to day 245, which was the persister isolate. Essentially, the accessory genome showed very little variation in genetic content, and there was no plasmid acquisition or evidence of incorporation of viral DNA within the bacterial genome. And what we do see is, again, this is the start of antibiotics in green, the start of phage in red, and kind of what diversity was present starts to disappear with antibiotics, and certainly no increase in diversity with administration of phage. So these later isolates then were retested for phage resistance, and we can see all the way out to 245 days following the start of treatment, there's been no development of phage resistance. So in conclusion, a longitudinal genetic analysis of 40 M. abscessus isolates over nearly 5 years was consistent with previous M. abscessus studies, suggesting a primary clonal infection with the emergence of adapted sublineages over time. For approximately 3 months after initiation of successful phage therapy, only isolates from the sublineage C were observed, suggesting that the phage cocktail was was more efficient at killing specific subpopulations. The one isolate in sublineage B that was recovered after 5 months of treatment or 5 months of negative cultures did not show an increase in antibiotics or phage resistance, suggesting that it may have aided phage treatment through mechanical sequestration rather than adaptive genetics. And overall, we did not see a trend towards greater antibiotics following the greater antibiotic resistance following the initiation of phage. And we also did not see emergence of antiphage neutralizing antibodies until late in treatment, and this was only partially reducing the phage activity. So I'd like to end here and thank the staff at St. Joseph Hospital and the Respiratory Institute who cared for our patient, and then all the the physicians and team members of the Colorado Adult CF program, and finally all of our collaborators in Pittsburgh and National Jewish, Colorado State University, and members of the NTM Core Clinical Research Service. And thank you very much.
Thanks, Dr. Nick, for a fantastic presentation. We have a number of questions, but I'm going to start. What you've demonstrated in your case presentation is a tremendous response bacteriologically. It's surprising to me, however, that the response was so delayed before it was so abrupt. In those first 96 days, where 6 out of 7 cultures were positive, did you have indications that the bacterial bioburden was decreasing?
Yeah, thanks for that question. We do have quite a bit of evidence, both molecular and metabolic, that there was massive lysis of the abscesses, even within the first few hours. And it didn't really make it to this talk, but we've been following urine lipoarabinomannan, and that is a cell wall component of Mycobacteria, and it spiked up pretty early within the first several weeks and peaked at about day 44, and then fell to non-detectable. And within we have PCR analysis of the sputum that shows quite a reduction in burden even within the first day. And then so we also have a lot of antibody work that shows the antibody levels just plummeting within the first day. And we think they're binding with the cell wall components. So we feel like the lysis occurred very early, and that I think that the persistence of the culture just relates more to the heterogeneity of the lung, that there's just so many nooks and crannies that it just takes a long time for the phage and the different airways to reach a certain level of exposure.
Excellent. Lots of questions coming in. First is from Dr. Luke Hoffman. Amazing work. When the group at Pittsburgh identified candidate phage therapy, do they do any in vitro experiments to see how quickly resistance might develop in patients' isolates?
Yeah, and I don't really, I can't answer that 100%. What I've been told by them is that they really, really don't see development of resistance in abscesses. And the kind of assays they do a dilution of the phage, and then they do a lawn of the phage, and the phage is so effective it just kills everything. So I don't know if they've got into that kind of methodology, but it's just such a different mechanism that I'm not sure how that would work actually.
Are there changes in co-therapies you generally make in starting phage therapy, such as starting or stopping other antibiotics?
Yeah, I mean, we have, this individual was on 5 drugs at the time of the phage was started, and it turned out to be a really well-tolerated antibiotic regime for him. So we've pretty much left it alone. I think that there are, especially in the field of Pseudomonas phages, there can be very clever ways of manipulating the antibiotics, but we were basically just giving him all that we have, and we didn't change it. It's actually changed very little in the last year.
A question from Dr. Katie Heisert. Do you have any other patients lined up for phage therapy? What criteria do you use in selecting patients?
Yeah, so the phage therapy is dependent on having a phage, basically. So we've sent isolates to Graham. We have another subject that he's identified 1 lytic phage to, but we haven't initiated treatment at this point.
A question from Peter Jorth. Did the NTM strain have evidence of an active CRISPR system? How common are CRISPR systems in NTM that may lead to phage resistance?
Yeah, I don't know the answer to that. again, Graham's told me that, that he's had by far the most experience in Mycobacteria phages. In fact, he might have all the experience with regard to the Abscesses use. And he says they just don't really see the development of resistance.
A question from Luke Hoffman again. Any change in other clinical symptoms with phage initiation?
Yeah, I mean, it was interesting. In this subject, about 90 days after he got phage, he was approved for Trikafta through the expanded label, the December 2020 expanded label for rare mutations. And that approval was not based on clinical trials. That was all based on the in vitro FRT studies. And so he, throughout this, he's been very productive of sputum, and he continued to be productive of sputum after Trikafta was started. Right about the time that the Trikafta was started and his cultures started to turn negative, he did have a decreased rate of hospitalization that was quite dramatic. However, he never regained his FEV1. His lung function remained very low. And I should mention that since we recorded this case, he's actually been transplanted. So that's why I'm speaking kind of in the past tense. He's actually doing very well. He was transplanted and he's back home now. But prior to transplant, he really didn't have dramatic changes in his symptoms other than stabilization of his hospitalizations and maybe some better weight gain.
There's a question from Alan Schmalstig. Was there any change in Mycobacterium abscessus morphotype over time, i.e. smooth to rough?
No, this was, I think we had 71 isolates and 69 of them were rough. It turns out that phage does not really work with smooth. And we were wondering if we would, as maybe sort of a sign of resistance, we'd start to see smooth, but that never actually happened. He remained rough throughout.
A question from Rafael Hernandez. Can you comment on the background antibiotic regimen? What was he on at the time of starting phage, and did it change over the course of the patient's phage therapy?
Yeah, for the pretty much the entire time, he was on nebulized amikacin, omadacycline, tadazolid, rifabut, or bedaquiline, and clofazimine. He was on those were the 5 drugs he was on. And so that that was a really easy combination for him to tolerate because it was just 1 inhaled and 4 oral, and then the IV phage. So, he'd been on probably 16 different drugs over the course of his treatment, and we just kind of stuck with that because of operational, and those looked pretty good on his drug susceptibility. So it was pretty consistent, those 5 throughout his treatment.
How impactful do you think the Trikafta was in his microbiologic response?
Yeah, it's really hard to say. I mean, I think it probably helped. I mean, I think it would be naive for me to say it didn't help. We have a lot of microbiome data that showed increased diversification of the microbiome later on, which has been definitely associated with highly effective modulator use. I think in particular, I think it may have opened access to some of these environmental niches where the abscesses may live that it can remain dormant and sort of secluded for a long time, and hopefully the Trikafta made airway biology more accessible to antibiotics and phage, but it's something we wrestled with a lot. There was definitely clear indications that he was improving before the Trikafta was added, but the magic really happened right about the same time that Trikafta was added. So it's something that we've kind of thought about a lot.
Thanks for a fantastic presentation and for answering all our questions, Dr. Nick. Our last presentation of the morning is Dr. Jane Gross. Jane.
Good afternoon. My name is Jane Gross, and I'm from National Jewish Health. Thank you for joining, and thank you to the workshop moderators for inviting me to present today. I will be presenting healthcare-associated links and transmission of non-tuberculous mycobacteria among people with cystic fibrosis, otherwise known as the HALT NTM study. This presentation highlights the Colorado adult CF program experience. There are no relationships to disclose related to this presentation. Healthcare-associated transmission of non-tuberculous mycobacteria among people with CF has been investigated at CF centers worldwide with conflicting conclusions. Both outbreaks and pseudo-outbreaks have been reported. Healthcare-associated transmission of Mycobacterium abscesses has been reported across the globe. One study reports widespread global transmission of M. abscessus via person-to-person transmission of fomites and aerosols. Other studies suggest no evidence of patient-to-patient transmission. This map of the world demonstrates outbreaks attributed to patient-to-patient transmission with a plus sign. Centers with outbreak reports include Hawaii, Washington State, North Carolina, Australia, the UK, Sweden, Denmark, and the Netherlands. In contrast, pseudo-outbreaks are noted by a circle and include Spain, Italy, and the UK. The objectives of the HALT NTM study are to systematically investigate healthcare-associated transmission and/or acquisition of NTM, to determine similarity among respiratory and environmental isolates, and to compare home residence watershed mapping among people with CF having genetically similar NTM isolates and receiving care in a single center. Do we have an outbreak an outbreak at our center? First, let's look at possible modes of NTM infection transmission. The blue boxes represent individual CF care centers. The red figures represent people with CF with a highly related NTM isolate. The blue figures represent people with CF with an unrelated NTM isolate. On the left, you can see demonstration of direct or indirect patient-to-patient transmission. In the middle box is an example of NTM acquisition via a common source, such as through water or water biofilm exposure within the healthcare system. On the right is an example of NTM acquisition via a common environmental exposure completely outside the healthcare system. Now let's focus on the HALT NTM study. This study provides systematic, evidence-based approach to investigating potential healthcare-associated NTM transmission within a single care center. Genetically similar isolate clusters are identified via whole genome sequencing. And as previously described, thresholds of less than or equal to 30 single nucleotide polymorphisms or SNP differences in the core genome raise concern for a potential healthcare-associated NTM acquisition, possibly originating from patient-to-patient transmission and/or a common environmental source. Here is a flow diagram of the HALT NTM study overview. First, we start with a number of NTM positive people with CF in a care center. Then we exclude people with CF that do not meet whole genome sequencing criteria. And we're left with the NTM positive people that have undergone whole genome sequencing. With whole genome sequencing of the core genome analysis, NTM clusters are identified based on subspecies. Through integrated analysis, we perform traditional shoe leather epidemiological investigation of clusters in the healthcare system. Next, we investigate common environmental acquisition via sampling of the healthcare environment, as well as home of residence watershed mapping. Finally, we perform pan-genome analysis of the core and accessory genomes to determine strength of isolate relatedness. Now let's move to the Colorado adult program. We had 165 NTM positive patients between 2012 and 2018. 108 people with cystic fibrosis met whole genome sequencing criteria. Of those, 57 patients were excluded due to having 3 or less NTM cultures. Ultimately, 80 patients underwent whole genome sequencing, and 27 people with cystic fibrosis were identified among 11 NTM clusters. You can see the cluster breakdown in the blue box. There were 6 M. abscessus clusters, 2 Mycobacterium intracellulare clusters, 2 M. avium clusters, and 1 Mycobacterium abscessus neciliens cluster. Phylogenetic trees comprising locally identified clusters among people with CF at the Colorado adult program are shown. Local and global reference isolates are shown as gray circles. The 11 sets of clustered NTM isolates are represented as colored circles, with each letter representing a cluster, A through K. The number in the cluster identifies the subject order based on first positive culture. The 6 Abscesses clusters are shown in panel A, with 4 clusters in the previously described dominant circulating clones, enlarged for detail. Panel B shows 2 M. avium clusters. C shows the Myceliens cluster, also falling within a previously described dominant circulating clone. And D shows the 2 intracellular clusters. Moving to the epidemiological investigation, this is a timeline overlap analysis of people with CF identified in NTM clusters. Clinic visits are open circles, hospital days, closed segments. Within 24-hour clinic overlap, vertical dashed lines. Within 12-hour clinic overlap, vertical solid lines. And hospitalization overlap is shown as vertical purple bars. Subject NTM status is also represented. Smear and culture positive is a red plus. Smear negative and culture positive is a yellow plus. Smear and culture negative is a blue dash. The letter again identifies the cluster and the number identifies the subject order. There are several episodes of smear positive subjects having hospital overlaps with culture negative subjects who later convert to culture positive after the opportunity for healthcare associated transmission. Another cluster demonstrates prolonged hospitalization overlap, with one subject being culture positive throughout the admission, and the second subject converting to culture positive during the admission overlap. Clusters that did not have overlaps are not shown. Next, we investigated sources of common environmental acquisition. First, through healthcare environmental sampling for NTM to look for common source exposures in the healthcare system. Second, we performed patient home of residence watershed mapping, and I'll show this in more detail. Let's focus on healthcare environmental sampling. We collected dust and water biofilms throughout the healthcare environment. A total of 161 healthcare samples were collected from the clinic and hospital settings. The diversity of environmental NTM is shown in the pie graph. There were 7 NTM species identified in the healthcare setting. Notably, the only species identified in the environment that was also represented in a patient cluster was Mycobacterium avium. We compared the environmental M. avium to all respiratory isolates and found no similarity among patients, either in an M. avium cluster or among any patients with CF and respiratory M. avium at our center. To further investigate common environmental acquisition of NTM, we performed patient home of residence watershed mapping. Panel A1 shows the relationship of the state of Colorado within the US. The state of Colorado is enlarged to show cities and counties within the state. Panel A2 shows the state of Colorado, but with watershed boundaries shown as light gray lines. The close-up in A3 demonstrates home of residence mapping of people with CF in the Abscesses clusters. All 3 subjects within cluster E live in the same watershed, which is highlighted with the gray background. Notably, cluster E also lacked any evidence of healthcare-associated overlaps. The remaining clusters, including intracellular, Myceliens, and avium, did not share a home of residence watershed, and these data are not shown. This slide demonstrates integrated pan-genome analysis comparing core genome SNP differences between pairs of NTM isolates within clusters on the x-axis versus percent shared accessory genome on the y-axis. Abscesses clusters are represented by circles, avium triangles, intracellular diamonds, and Myceliens by an upside-down triangle. Pairs of subjects with observed healthcare overlaps are noted by a solid square around the symbol. In the upper left-hand corner, we highlight cut points of less than or equal to 10 SNPs and greater than or equal to 95% shared accessory genome in the dashed line box. This dashed box represents the most stringent level of clustered isolate similarity. Keeping your attention on that dashed box, M. avium cluster D demonstrated sequential healthcare associated opportunity for transmission from subject 1 to 2, and subject 2 to 3. And they maintain cluster isolate similarity at the stringent pangenome level. 2 members of Abscesses cluster E showed no evidence of healthcare-associated opportunity for transmission, but resided in the same watershed and have 0 SNP differences at the core genome level, and then greater than 98% shared accessory genome. Abscesses cluster K showed no evidence of healthcare-associated opportunity for transmission, nor did they live in the same watershed, but still showed a high level of similarity at the pangenome level. In summary, the presence of genetically similar isolates is insufficient by itself to demonstrate healthcare-associated NTM transmission. The HALT NTM study provides a framework to standardize epidemiologic investigation for potential healthcare-associated NTM acquisition and/or transmission among people with CF. HALT NTM couples traditional shoe leather epidemiologic investigation with healthcare environmental sampling and home of residence watershed analysis. This will improve understanding of the frequency and nature of healthcare-associated NTM transmission among people with cystic fibrosis. This process also has the potential to identify where acquisition might be at higher probability within the healthcare system. Our center investigation of the Colorado Adult Program suggests that healthcare-associated transmission of M. abscessus is rare at our center. We report likely healthcare-associated transmission of M. avium, supported by evidence that both clusters demonstrated biologically plausible episodes of healthcare-associated opportunities for transmission. Additionally, one cluster met stringent threshold for relatedness with less than 10 SNPs and greater than 95% shared accessory genome. And the second cluster differed in criteria only by 11 SNPs. One M. abscessus cluster, in which patients resided in the same watershed, revealed no episodes of healthcare-associated opportunities for transmission, but genetic similarity at the core and accessory genome support environmental acquisition from a common location. 2 M. abscessus clusters demonstrated episodes of healthcare-associated opportunities for transmission, but they did not meet the stringent threshold of isolate similarity to support healthcare-associated transmission. The remaining clusters did not share opportunities for transmission, nor did they live in the same watershed. The HALT NTM study is open for enrollment, and we have multiple concurrent parallel CF center investigations underway. Thank you to the Cystic Fibrosis Foundation for providing the funding to make this work possible. Thank you to the large team of collaborators that carried out the work presented today, including the team at National Jewish Health, the support from St. Joseph's Hospital, the Colorado Adult CF Program team, as well as the CF patients and families who participate in research trials. I look forward to any questions you may have. Thank you.
Thanks, Dr. Gross, for such a fantastic presentation. And certainly on a personal note, thanks for really advancing the idea that a robust framework is required for understanding infection transmission studies, and that it's not simply genomics, but linking that with good old, as you say, shoe leather related epidemiologic investigations. There's a number of questions from the audience. I'll start with a question from Dr. Katie Heisert. Do you think NTM is spread person to person or via fomites? And what are the implications for our clinic workflow and work practices?
Thank you for that question. There are studies that suggest that NTM is spread by fomites. I think in our investigation, at least of our center, we feel like, we did this epidemiologic investigation and we looked really closely at where patients had overlap. Not only did they overlap in the clinic setting, but where in that clinic setting could they have possibly overlapped? And it was difficult actually to pin that down. And of course, this is all retrospective. So we don't have, a smoking gun, if you will, of a surface to swab immediately followed by an infection. So it seems in our mind that it's likely indirect patient-patient transmission, where the surfaces, have NTM in them. And we really think that the biofilm, the water exposure through water biofilms is a great place to look, because we know that lots of water biofilms are colonized with NTM, and that's why we focused our environmental sampling there as well as in dust. But notably in our center, we didn't find, any clinically relevant NTM other than M. avium, and that M. avium ultimately did not match any of our patient samples. So it's a hard question to answer, in part because it's, NTM is so slow to grow. So to even discover it in a patient takes weeks to months. And by the time you recognize an epidemic, you're recognizing this, or an outbreak, if you will, you're recognizing it well after the fact of the initial infection acquisition. So this is, very retrospective. So I think it's a difficult question to answer, but a great question. But I do think it's more indirect patient to patient, or just coming from the environment.
Thank you. Question from Luke Hoffman. A very promising framework. At this point, from what you've found to date in published works, what do you feel is likely to be the most common mechanism of acquisition of mycobacteria, if there is one?
That's a great question. And again, I think, the literature historically has said you acquire NTM from your environment, and that can be from soil, from water exposures, from lots of different places. It is very clear from the literature that there are these dominant circulating clones that are readily identifiable, both, in the US and overseas. And so it seems these dominant clones are probably just dominantly endemic in the soil and water supplies. And likely patients are picking them up from these, common organisms are picked up from commonly found organisms in the environment. I think there was a second part to that question that I'm potentially missing.
If you believe there's the most common mechanism.
Right. So I think likely it is probably from environmental exposures. Our study is limited to looking at environmental exposures within the healthcare system. It would be nice to potentially expand this to outside the healthcare system, because obviously patients are spending a lot more time in their home and work environments than they are, in the healthcare system.
Another question related to infection transmission. In your investigations of whether there are overlaps in hospital and clinic, did you identify if there were breaks in institutional standards of infection control or at that time?
That's a great question. So we did look at that as part of our retrospective epidemiological analysis, whether or not the standard CF Foundation recommended infection controls were in place. For example, gowning and gloving for all patient interactions, limiting patients in common waiting areas, things like that. And so I think our system in the timeframe we examined was pretty consistent throughout that timeframe from 2012 to 2018. Certainly in the hospital setting, we were working in a new hospital setting during that time frame, where our patients had been cared for at one hospital and were transitioned to another. And this is a very modern, up-to-date hospital with very good infection control practices. Also, we have a dedicated CF floor in which everyone that works on that floor is very accustomed to the CF infection control measures. And so I think, at least in the hospital setting, that was very well adhered to. In the clinic setting, we did have a small transition during that time frame from initially just physicians gowning and gloving with patients, and then there was a change to implement all providers, regardless of your interaction with the patient, would gown and glove. So that was one transition that did happen during that abstraction timeframe that we looked at, but it didn't seem to have any direct consequence on our findings of potential transmission.
And were there any investigations of whether patients had personal relationships outside of healthcare encounters?
Also, another great question. We did want to address that. So the way we address that without, having to go to each patient and ask them to reveal, do you have contacts, which is a little trickier, right, to do. We really wanted to do this through investigation of what was available in the healthcare system. So we were well aware if patients cohabitated, for example, with another CF patient, either in the form of siblings or roommates or spouses. So we did have that level of information. And we had the level of information that was available from the EMR. If patients, offered up, other contacts, then we would know that from the EMR, but we did not specifically ask patients, directly who did they have contact with. And as far as we could tell in our evaluation, we did not have any patients that were siblings living together or, married to another person with CF. So there were no clear exposures from our investigation.
There's one final question from Dr. Hoffman again. He was also wondering how the SNP-based cutoff for relatedness was defined and whether it differs in different subspecies or lineages.
That's a great question. So for Mycobacterium abscessus, all subspecies, the core genome SNP threshold is 30 or less. For avium, it is 25 or less. And it may seem odd that there's a difference cutoff. The Mycobacterium abscessus have been studied the most as far as transmission goes, both in our center and in others that have reported concerns for outbreaks. And 30 is consistent with what other outbreaks have been reported. So that also is consistent when we look at a single patient that's infected with a Mycobacterium abscessus over time. There does not seem to be a highly variable rate of mutations within a single patient. So that makes sense for that cluster analysis. And then for abscessus, there does seem to be a higher mutation, for avium, there seems to be a higher mutation rate. And based on our own internal data of looking at individual patient isolates over time, 25 was the better cutoff for the avium species.
Excellent. That's fantastic. Thanks so much for a great presentation and for answering all our questions.
Thank you.
With Dr. Gross's seminar closing, I'd like to thank everybody for attending today's talks. They were certainly fantastic and have certainly brought us a lot to think about. If anybody has further questions for any of the expert panel, I'll ask the presenters each to hold on for the next couple of minutes such that we can convene a joint panel if there are general questions. And if there are not, we'll give everybody a few minutes back in their day. So we'll just ask you to hang on for 2 minutes. And if questions are coming through, we'll let you know. And if not, everybody will be free.
We are getting kudos in the chat for a great session.
Looks like all the questions have been answered. So on behalf of myself and Katie, thanks everybody for a great day and great talks. Really exciting stuff.